Hi class, in this recording we're going to focus on your eye and structures of vision. This is going to be an exercise 23 of your lab manual. We're first going to focus on the accessory structures, then we'll dive into the layers, and then we'll focus on cavities of the eye. So let's look in visible body here. We'll pull up visible body, and just to give you some reference point, I clicked on the microanatomy view and then the eye. So this shows all of our accessory structures of the eye, and I like this because it gives us a good overview of what we're trying to look at. So as we look at our lab objective list, we first have the conjunctiva. The conjunctiva itself is not visible here within visible body. It's the membrane that holds our eye into the orbital cavity, or helps connect our eye to the inside of our eyelid. So I'm just going to do a quick image search of the conjunctiva to really emphasize what is the conjunctiva, and you can see it right here beautifully. If you just invert your eyelid, you can see this pink tissue on the inner lining of your eyelid. That pink tissue is your conjunctiva. I included conjunctiva on your lab objective list because of a common diagnosis called conjunctivitis, which is commonly referred to as pink eye. Hey, speaking of which, here's an image of conjunctivitis, inflammation of that membrane that connects the sclera to your palpebrae, or eyelid. So as we go back to our terminology list, let's look at the lacrimal apparatus. This is responsible for washing our eye, producing and washing our eyes with tears, and then removing the tears from our eyes. So when we look at this structure, we'll just go over it in the path of tear production. And there's a view here that better represents the lacrimal apparatus as a whole. We first have the lacrimal gland. This is going to produce the tears. And the pathway of travel here, to give us a frame of reference, let's go ahead and annotate the screen here. I'll use green again because it's my favorite color. So we have medial, and then here on this side we have lateral for our frame of reference. And the general pattern is that tears will travel medially and inferiorly across our eye after being produced. So we have up here the lacrimal gland, which makes the tears. We are not going to ask you to differentiate the lacrimal gland subsections. Just know the lacrimal gland as a whole. And then we have the lacrimal caniculus. The lacrimal caniculus are these tiny passageways that will carry excess tears away from the eye into the lacrimal sac. And this lacrimal sac is located directly above the lacrimal bone of the skull. And the nasolacrimal duct is a tube or passageway through that bone that is a passageway that goes through that lacrimal bone. And that nasolacrimal duct, as its name implies, if we look at those rote words, naso meaning nose, lacrimal referring to the lacrimal gland, it's going to carry secretions from the lacrimal gland, tears, into the nasal cavity. So if you think about when you hit yourself in the nose, or maybe you're going through some emotionally volatile situation that causes you to tear up, the reason you start to get sniffly when you start crying is because your lacrimal gland is producing an excessive amount of tears. Those tears travel across the eye, travel through the caniculi, to the lacrimal sac, and then through the nasolacrimal duct down into your nasal sinuses or nasal passageways. In other words, you're draining a lot of extra fluid into your nose when you start to sniffle or when you start to tear up. So that's the lacrimal apparatus. Let's talk about the muscles of the eye, the extrinsic eye muscles. And if you remember your directional terminology, you already know all of these muscles. Lateral, medial, superior, inferior. I mean, it's just beautiful. The names are just so easy. So we have a directional term. Lateral means towards, away from the midline. Medial means towards the midline. Superior means above. Inferior means below. When we see rectus, I want you to think of muscle fascicles that are in a straight line. And when you see oblique, I want you to think of muscle fascicles that are not in a traditional plane of a body part. And you'll see what I mean when I show them invisible body. So we'll back up here and just do the whole view of the eye. So we have the muscles visible. Here on the superior margin, we have the superior rectus muscle. On the inferior margin, we have the inferior rectus muscle. Notice those fascicles are all parallel with each other. On the lateral margin, we have the lateral rectus. On the medial margin, we have the medial rectus. You guessed it. And then let's talk about the obliques. And if I zoom out a little bit here, the inferior oblique muscle right here is at an awkward angle as it crisscrosses over the eye. And then in the superior oblique is just messed up. It actually has a pulley system, a biological pulley system built in. So right here, the superior oblique muscle goes up to here, and then there's a hook of connective tissue in the eye orbit that it goes through and then goes back towards the optic nerve. So that superior oblique muscle, that's a pretty unique muscle. Not many muscles in the human body have a biological pulley system built in. Let's unhighlight that and go back to our lab objective list. So those were the muscles of the eye. Let's move on to layers of the eye. And we can go, let's go with this view, clean it up a little bit for us here. Those blood vessels tend to be kind of messy. So as we look at our list, we have the external layer, and it's going to be made up of the cornea and the sclera. The cornea is the most anterior portion of the eye and is 100% transparent. So if we were to look at the view here, a lateral view, I can click on the cornea as that most anterior layer. The sclera is commonly referred to as the white of your eye. So in this view right here, I'll go ahead and zoom out. Here we have the sclera right there for us as the whites of our eye. Notably, I want to emphasize how the sclera goes all the way around to the posterior margin of the eye. It wraps around the entire surface of our eye. So those were the external layers, the cornea and sclera, and then we have some middle layers. The choroid is a dark, highly pigmented membrane that wraps around the eye. It serves the function of keeping unwanted photons of light from getting into the eye. So here we have the choroid that is deep to the sclera and lines the sclera of the eye. It goes all the way around the eye. And then deep to the choroid, we have the retina on the posterior half of our eye as the innermost layer. On the anterior, where there is no choroid, we instead have the ciliary body. So as I look over here, I'll just go ahead and zoom in right through the back of the eye because we can do that with this software. That's pretty cool right there. So here is a view from the interior of the eyeball looking towards the anterior. And we can click on, oh, come on, let me highlight it. There we go, our ciliary muscles. Let's use our breadcrumbs here. Ciliary body. There we go. We highlighted the whole thing now for us. So let's zoom back in. Here we can see the entire ciliary body highlighted. This ciliary body serves the function of anchoring and changing the shape of the lens of the eye. And it's broken up into ciliary muscle, ciliary processes, and suspensory ligaments or ciliary zonules. So as we look at the ciliary body, that's the whole kit caboodle. And it's really not going to let me click on that. That is frustrating. So we have the ciliary muscles that make up the ciliary, most of the ciliary body. And then we can see these ridges. I'll go ahead and highlight a couple of the ridges in green. Right there, right there, right there. Those ridges are the ciliary processes. Those ciliary processes are important because they serve as the anchor points of these small, teeny tiny ligaments. So that's still not letting me highlight them. That's frustrating. We'll skip those processes. So the ciliary processes serve as the anchor points of these tiny ligaments that connect directly to the lens of the eye. The name of those ligaments that connect to the lens of the eye are the ciliary zonial or suspensory ligaments. I'm a bigger fan of suspensory ligament just because it's easier to spell, particularly in a lab exam situation. Let's back up a bit. There's an asterisk right there next to choroid. So we need you to be able to recognize the choroid from a histology view. So I'm going to go ahead and just click on, let's go with this eyeball right here. This is a beautiful eyeball. And when we're looking at this eyeball, I think this is probably a Norwegian rat eye, if I had to guess, or maybe a monkey eye. We can see our choroid here as we look at the histology. So on the back of the eye, there's this layer right here that's highly pigmented, that has a lot of black pigmentation to it. That's the choroid. And since we have an asterisk on your lab objective sheet, we want you to be able to recognize the choroid on a visible light photomicrograph. Let's talk about the iris and lens themselves. We can see the iris and lens, or the lens at least, right here in this photomicrograph. Not that you need to know the lens on a microscope slide, but hey, here it is. This is what the light will travel through as it enters our eye. And you can also see the iris itself as well. The iris is visible as this dark pigmentation that goes right up to the lens. So on the superior and inferior margins, you can see the iris. And there's an opening in the iris. I'm highlighting that opening in red. That opening that allows light to go to the lens is referred to as the pupil. So here in Anatomy Atlas, we can see the iris as the colored portion of the eye, and in this model, it's a brown iris. And then there's an opening, a black circle. That black circle, let's go ahead and zoom in. Right here, that represents the pupil. And then if I look at the posterior here, it's still not letting me select the lens, but we can see the lens as this large circular disc within our field of view. So those are the layers of the eye. Let's focus, or the middle layer, let's focus on the inner layer in more detail. In our model here, I've already showed you once, but it's worth reviewing, just refreshing. We have the retina. The retina is the deepest layer of the eye, and notably the retina is going to contain our photoreceptors, our rods and cones. So if we go to this view right here, here we go, we're in full screen. It kind of looks like a 7-layer salad to me when I look at the retina in the back of the eye. So I'm just zooming in, taking my time, happy little retina. And we can see here the 7-layer salad. That is our retina. So I went ahead and put a red block around it. So this layer right here that alternates between pink and purple banding is the retina. And I want you to be able to recognize the retina on a microscope slide because it's just crystal clear. Our next layer, let me clear that annotation off for us, is the optic disc and the optic nerve. And as we look at the retina, let's go ahead and zoom out. there's a region of the retina that does not have any photoreceptors. And it's notable because there's a total lack of purple nuclei. This area right here of the retina doesn't have any of the purple nuclei. So right over here, above and below, we see the purple nuclei of the retina. And then right here in the middle, in the red rectangle, we don't see those layers of purple nuclei. That means there's no photoreceptors in this area. This area that I've blocked off is called the optic disc. And you can see it quite clearly in this photomicrograph. And then if I were to back up here, this large band of tissue that extends from the eye, from the optic disc, is referred to as the optic nerve, cranial nerve 2. Now, something else that we want you to be able to recognize is the optic nerve and these structures in visible body. So I'm going to go ahead and go back to visible body just to emphasize these structures. Here is the optic nerve, and directly anterior to the optic nerve, we have this circular structure right here. That's our optic disc. And then off to the side, just want to make sure, yep, off to the side, we have another circular structure. This circular structure right here has darker pigmentation. It's the darkly pigmented region in the back of the retina. This is referred to as the macula lutea because it's a darker yellow color. And in the very center of the macula lutea, we have a small indentation. And that small indentation in the very center is referred to as the fovea centralis, the innermost part of the macula lutea. A lot of times you'll hear of people that are losing their vision because they have macular degeneration. Macular degeneration is when we have breakdown of the cells that make up the macula lutea, and that's a very serious issue. And then finally we have some cavities of the eye. When we think of cavities of the eye, they're just broken up into segments. Let's go ahead and pull it up here. We have a posterior segment of the eye. This posterior segment of the eye is going to be posterior to the lens of the eye, as it's defined in most anatomy texts, and it's filled with a jelly-like fluid. So the space is the posterior segment. So if I was to ask you what is the space, I would want to hear posterior segment. If I was to ask you what fluid fills this space, I would want you to say vitreous humor. Humor is an old school term that means liquid. You maybe have heard of how in medicine, particularly in medieval times, with medieval medicine, they focused on balancing the 4 humors of the body, and that's when you would look at blood, bile, saliva, and urine and try to get the ratio correct to heal somebody. Usually that just meant you cut them and let them bleed a lot. And we don't do that anymore, generally speaking, in healthcare, which I think is a good thing. But that's why this is an archaic term. Humor is an archaic term for liquid. So we have the vitreous humor in the posterior segment. In the anterior segment, we have the aqueous humor. And that anterior segment is going to be anterior to the lens of the eye. So I'm going to back up here to show these segments in a little bit more detail. So here where I'm putting the red X, this is the posterior segment, and then in green, I'm highlighting the anterior segment between the lens and the cornea. The lens is this dark pink ovoid shape, and then the cornea is this lighter pink ellipse, or we'll say letter C on the anterior of the eye. That's all we have, class, for structures of the eye. If you have any questions or comments, please feel free to post them in the class discussion board or to shoot me an email. And as always, happy studies.